Okay, so we'll get started here in a few minutes. How was AMP2? Like, how many times did you have me slowed down or not able to hear me?
Dr. Ong,
Yeah.
is there slides for today that I can look at on my phone while we go over the notes or something for today?
I'm gonna share. I'm gonna be sharing my screen.
Okay.
That's how I'm gonna do it. Where sometimes I will show me like I am now, my face, and sometimes I'll be sharing my screen and it will be the slides, and I'm able to write on the slides kind of messily.
Okay.
But that's how it'll go.
Okay. Dope.
Constant, are you on now? What does this mean? I can see your initials, CT. Let's see. Let me try that again. It says calling. See, Samantha. Okay, Constance on. Great. Let me update that in Portal. And we'll be ready to get started. Okay, I am recording this in case you all need to watch it later, or if people weren't able to join at this time, they can join at a later time. So what I'm going to be doing today is sometimes I will talk to you on the camera like this, and sometimes I'm going to be sharing the screen. And on my screen is going to be the PowerPoint slides like I would usually use. And I will be able to write on them some. I'm probably going to go slower than I usually would in class. And that way, if my sound glitches out or something freezes up, I won't be too far ahead when it comes back in. Yeah, Samantha? Somebody, yeah.
Hey, Dr.
Hey.
So I just want to let you know that me and Brooke were both watching from my phone. So
Thank
she just
you.
wanted me to let you know that we're both here.
Thank you. And
Okay.
I will update that attendance in portal.
That's too much for that context. Okay.
But this summer, Han and I would always be on faculty meeting, and somebody, are you both there? Like, yes, we're. So, yeah, that's smart and good. Okay. It also, anytime that you can't hear me or if you can't see what I think that I'm showing, just like burst in here with a comment, or you can also put it in the chat box. Sometimes it might take me a little bit to see the chat box. So I'm going to share my screen now. And go to my slides, and this is in the middle of the slides for today. So I'm going to get this up on full screen, and then once in a while I will leave this screen and just go and check the chat box, or I will stop sharing screen and make my face come back and look at you so that it doesn't get boring and monotonous. But what we're going to talk about today are microbial enzymes that allow microbial metabolism. And this is probably one of the topics in the class that doesn't always seem obvious why you should care about microbial metabolism and enzymes. But it's really important to the ways that microbes infect us and the ways that we can kill them. So that's why to care, and I will be highlighting some of those things as we go. So this particular picture that your book uses to describe metabolism, I really like it. I really like this because it's showing a lot of different processes that we're going to talk about throughout this whole unit in a pretty good visual form, but not too complicated. So what this is trying to show is that microbes in some way are going to have food. And this writing is going to be so slow and kind of silly because I'm doing it on a trackpad. So in some ways, any life form is going to have a food molecule. And the ones that we talk about that matter to human health, we're going to pretty much assume that the food is a sugar. That word is supposed to be sugar. And the microbes are needing that fuel, like glucose is a sugar, so that they can get energy out of it. So they're going to use it for energy. And they are going to use it for their carbon source. So this says carbon, energy and carbon. And the microbes that we care about medically, they all are chemoheterotrophs because they are using some kind of organic food molecule for both energy and carbon. So when they break down their food, we call that process catabolism or catabolism. And you have learned about catabolism before somewhere when you learned about aerobic respiration, these three things. That's what we will talk about on Friday, as well as fermentation. So these processes that we talk about Friday have to do with how do microbes use their food to get energy out and carbon building blocks. These molecules are intermediates along the way. So these are some important chemical intermediates that microbes produce while they're breaking down their food. And just trust me that that says intermediates. And then they will further break down those intermediates into building blocks that they then can use to build more microbes. So you kind of break down food to produce energy and building blocks, and then you build the building blocks into more cells. So when we talked about how many cells, and we had that equation N sub F, well, it's hard to write, N sub F equals the initial times 2 to the N. Hilarious writing. And we talked about when conditions are good, they will multiply quickly. You have to have enough food in the environment to build more cells. So I really, really like this picture because it's referencing so many levels of microbial life. And you'll see me come back to this again in the upcoming slides. So one thing I want you to have really firmly in mind from this class is catabolism versus anabolism. So catabolism is a breakdown process where you are breaking down a big molecule into something smaller and you get energy out. And then you use those pieces and those that energy to do anabolism, which is building up. Building up. You have to have the food to have energy to build up. You have to have the food to have building blocks to build more of you if you are a bacterial cell. In this class, I focus a lot more on the catabolism part. I don't go into much detail describing the building up processes. We spend more time on the catabolism, but breaking down food and getting energy, that's a catabolic process. And all of these processes are using enzymes to get these things to happen in a timely manner. So I want to go check the Teams chat here, and if you have, if my sound is slowing down or you can't see something, always let me know. I'll just check once in a while. That's working. Okay, so I think you had a quiz question today. What color are the enzymes in your book? And the answer is purple. They are colored purple. So we have here a picture of some kind of random enzyme that has a job to take these two substrates, these two chemicals, and if it can act on one of them, it can turn one of them into a product. So we see here that for this enzyme, this is the substrate that fits with it. And those two combine together, and this enzyme will catalyze. What just happened? I don't want to do that. I want to write the word catalyze right here. This enzyme is going to catalyze, this is a Z, the reaction of taking this substrate and breaking it apart into products. So this particular reaction that this one is doing is catabolic. This enzyme is performing a catabolic function. This substrate could technically fall apart into these two products and have that happen spontaneously. This is something that could happen without an enzyme, but if the enzyme is there, it's going to be a lot more likely to happen. So that's what this is referring to, that we need this enzyme to speed up this reaction that could happen anyway, but the enzyme is going to make it work faster. And the way that the enzyme does this is by positioning this substrate in a way that makes it more likely to break apart. All of this, gotta activate the pen every time. All of this depends on the 3D shapes. All of this depends on the enzyme having the right 3D shape and the substrate having a complementary shape. If this is going to happen, this enzyme has to have the correct shape. And the shape of the enzyme comes from a protein folding into the correct 3D shape. And that information, we talk about that next week. We talk about transcription and translation of proteins. But if the enzyme is going to do its job, it has to fold into the correct 3D shape. How does this seem so far? Go check the chat box. Okay. Seems okay. Full screen. There we go. So I want to put here some examples of enzymes that we have talked about in this class or that we will talk about in this class that matter to human health. So one example of an enzyme that is needed by microbes to do important jobs is catalase. You remember that one? Catalase is one of those enzymes that has to detoxify the reactive oxygen species. So that's an example that if this enzyme was catalase and this is peroxide, this enzyme is supposed to turn the peroxide into oxygen, that's oxygen, and into water, and detoxify a toxic substrate. So if catalase is going to do this, it has to have the right shape. If it doesn't have the right shape, the dangerous peroxide over here would stay dangerous and cause problems for the cell. So it's really important for the cell that the catalase enzyme would be properly to do this job that the cell needs done. One example. Another example, write it in different color, is an enzyme that would break down antibiotics. This would be really helpful if you were a microbe and you had an enzyme that would break down antibiotics. Whoops. Antibiotics. I can't dot I's in this system. But if this were now a drug, an antibiotic drug that's going to kill the microbe, but the microbe has an enzyme to destroy it, that's really good if you're a microbe. So having that kind of enzyme would be really helpful. The third type that I'm going to use as an example is something that we will see in lab pretty soon, which is called urease. Urease. This is U-R-E-A-S-E. Urease. And what urease does is it takes urea, like in our urine, and lets the microbe break down urea and use it as a food. So really what urease allows is for a microbe to eat urea as a food source. Most life forms cannot do that, but there are some microbes that love to have urea for their food, and that makes them really good at causing urinary tract infections, or UTIs. Urease is an enzyme that lets a microbe use urea as a food. I hear somebody. Yeah.
Yeah,
Go ahead.
Dr. Long. Did you give a name for the microbes that can break down the antibiotics, or is that just a vague term right now?
So if you're a microbe and you can break down antibiotics, we would say that you are antibiotic resistant.
Okay.
And I don't have room to write that on here, but if you have this capability, you are antibiotic resistant. And we talk about that a lot at the end of the semester.
Okay. Yeah. I was just curious to see if there's an because you put like catalase and then urea for the other one, but then it's kind of just vague.
The name, like the names of the enzymes that do this are extremely long. So that's why I'm not.
Let's skip that for now then.
Yeah. I would not be able to make it legible with this style of writing. But they do have names, but I can't write them in this format.
Okay, sweet. Yeah, I was just curious about that.
Yeah, thanks for asking a question.
Oh, yeah, thank you.
It makes it livelier. Okay. So enzymes are really important for all kinds of things that cells do. Once in a while, the protein part of the enzyme is going to need, of course, there's no purple. There's no purple pen. But this is close. The purple part, the protein part of the enzyme, is not enough to get the job done. It's important and it's necessary, but it's not sufficient to do the whole reaction. So in those cases, when enzymes need a helper like a metal, and an example of that metal might be iron, which we would write like this in a chemical sense, an iron atom right there, that might be a cofactor. Or a coenzyme like, shoot, what is this called? Oh, yeah. I remember what I wanted to say. Folic acid. Folic acid. To us, this is a vitamin. We call it a vitamin, but it's needed as part of a bigger enzyme to let the enzyme break down food for energy. Microbes also need folic acid, and they need that to participate with an enzyme to break down food for energy. You might have some enzymes that need both a coenzyme and a metal cofactor. Let me say a little bit more about how these two are different. If something is a metallic cofactor, it's one atom. It's one atom of a metal. So like iron or zinc or cobalt or something like that. Magnesium. It's just one atom of a metal. If something is that's implying that it's a big molecule. That says big molecule, not just one atom of a metal. Once you have the protein plus this other thing, it's called holoenzyme. Holoenzyme. So we've got this word holoenzyme. Write this so slowly. The holoenzyme equals the protein part, which is apoenzyme, plus one of these two. So this picture is showing you that the part of the enzyme that's just the protein in purple, that's apoenzyme. But if they're supposed to also have one of these other molecules together, the apoenzyme plus the cofactor, we would call that the hollow enzyme. The whole thing. That's how I remember this. Whole thing, hollow enzyme. That's the part that could do the function. So if you're a microbe and you make just the protein part of this enzyme and you don't have folic acid, whoop, no folic acid, I don't have it. It doesn't matter that I have this. Folic acid is essential for this to work. And if I don't have folic acid, I'm going to die. That sounds really drastic, but there are some bacteria that we kill them in the body by messing up their enzyme so that they don't make folic acid. There's certain antibiotic drugs that mess up folic acid production so that the bacteria can't make the proper holoenzyme and they're going to die without the folic acid. That kind of thing is why I spend a lot of time talking about enzymes, because let's go backward. If you mess up an enzyme, pen, I'm going to go backward and I want to use the pen. There we go. If you destroy the function of an enzyme that's essential to the microbe, you can kill the microbe. So this is probably worth writing. If we kill an enzyme or we prevent the enzyme from functioning, that's basically the same thing as killing the cell. And most drugs that we use to treat bacterial infections are doing something to kill an enzyme. We learn about that later in the semester. Okay, so I want to put my face back on the screen here just to switch things up. I haven't had as many interruptions in this class. I had some weird interruptions in AMP2. I had a package delivered, and then one kid decided to sweep the porch while I was out here, which is nice, but kind of distracting. So go back to share screen and keep talking about enzymes. There we go. So the next, I do not love how your book handles the rest of the pictures in this chapter. It's really important concepts. I don't love the pictures, and I don't really like how they organize the reading. So this would be a good time for me to say that for Friday's lecture, I don't want you to do book reading for Friday. That's not what the reading assignment is. But for Friday, I'm going to want you to watch videos. So Friday, make yourself a note. Before lecture on Friday, there are video assignments to watch. And these are linked in the page that goes with anaerobic and aerobic metabolism. What these videos are these are from a class that I used to teach that's the non-major bio class. And they're about glycolysis and Krebs cycle and electron transport chain at a really introductory level. So instead of you having to try to read this stuff in the textbook, I would rather you watch those videos. They are kind of long compared to doing the reading, but I think that they're really better than doing a reading for this topic. So that's what the assignment is for Friday, is the videos. And the reading quiz is about the videos. So make sure that you leave enough time to watch those. I think they're about an hour and a half together. I heard a Teams beep thing, so I'm going to check what that is. Great. Okay. So remember for Friday is to watch those. Okay, now that I have made that announcement, when you look at pathways like this, and this one is describing that there is a sugar, glucose, which is a fuel source for this microbe. It's got 6 carbons. And then there's an arrow here. And then there is a new chemical here. And then we see some arrows from this new chemical to these molecules called pyruvate. What I want to focus on in this picture is the concept that that represents an enzyme, that arrow. There is an enzyme that the cell has that turns glucose, a sugar. Oh, thank you. Need to share my screen. How about now? I saw the notification. It should be working now. Okay, so what I was trying to show is there is a chemical, glucose, that's a sugar, and cells can turn it into a different product called fructose 1,6-diphosphate. That's not the point of this. My point here is for all of us to recognize that this arrow right here implies that there is an enzyme that does that job. There's a particular enzyme that turns glucose into fructose 1,6-diphosphate. And that's an essential enzyme for this cell. It would have a name. In people, it's called hexokinase. You don't need to know that for this class, but that's a particular enzyme, and it has a particular shape, and it's doing this particular job for the cell. It's just implied by the arrow. So likewise, down here, there is a separate enzyme. This is a different enzyme down here that's doing the next step in the process. So we would call this one enzyme number 2. There. Stop doing that. I want to stay on full screen. So enzyme number 1, enzyme number 2, and so on. If you already knew that before this class, and I'm being so obvious right now, I don't mean to bore you, but I don't think that I got that when I was much younger. That means that there's an enzyme, and that means that there's a gene that produces that enzyme for that particular step that is used in this overall process of glycolysis. For E. coli, write this up here. Pen. So for E. coli, there are probably about 10,000 enzymes. This means 10,000, 10K. It takes about 10,000 enzymes to make an E. coli cell, roughly. And each one of those enzymes is pretty important to that cell, to its life. So out of all of the 10,000 enzymes or so that E. coli makes, there are some enzymes that it makes all the time, every day. Those we call constitutive. Those are constitutive enzymes. There's pretty much always a constant amount. So the enzyme here is shown in purple. There's always about a constant amount, no matter what's going on in the life of that cell. So I compare this to like when you go to the grocery store and there's stuff that you always buy. So for our family, it's milk. Like we don't question whether we're going to get milk. The question is how much milk do we need? Do we need 2 gallons or do we need 4? We hardly ever buy one because we have 3 boys and we try to get them to drink milk the most. And then especially where we've all been home this summer, we've just drunk the most extreme amounts of milk. So I would consider milk to be a constitutive grocery for us. We always need it. All the time, every day. There are some enzymes that a cell might make more or less depending on the situation. These are called regulated. So if an enzyme is regulated, it's something that you don't always need if you're the microbe, or you might want to change the levels of how much you need. So an example of this is hamburger helper. I'm just going to write HH. And yes, we eat hamburger helper quite a bit here. And my middle son told me yesterday that we are out of the good kind, which to him is the beef pasta. We're out of the good kind and we need to get more. So we don't buy that every time we go to the store. We tend to stock up on it, and then when it runs out, we can make some more. That would be a regulated grocery in our house. I'm trying to think. Here's an example. So catalase is kind of a constitutive enzyme. If you live in an oxygen environment, you need catalase all the time. But a regulated enzyme might be one that is an antibiotic breaker, something that breaks apart antibiotics. You don't need that unless there are antibiotics around you. You don't need to make that every day all the time. That would be regulated. Okay, next, super big concept. Super big concept here. We've been talking so far, or I've been talking at least, about the enzyme. Like, so there's the enzyme, and it's important, and we need the enzyme to run the reaction for us and turn the substrate into product. But the enzyme doesn't just appear magically in the cell. The enzyme is coming from genes that allow production of the enzyme. So if you're a microbe and you're going to make an enzyme, you have to have the gene or the genes in your genome to make that enzyme. It doesn't come from nowhere. So you've got to have the DNA to allow the production. So you have the DNA, then it's transcribed into RNA, then it makes the protein. The protein has to fold correctly into the proper 3D shape. Got to fold into the 3D shape. But none of this can happen if you don't have the gene. So if we think back to when we talked about structures, bacterial structures, the genes can be on the chromosome. Most of the genes that a microbe has are going to be on the chromosome. And if they're on the chromosome, they're considered essential genes. But the other place that there could be genes for a microbe is on the plasmid. Should be an I there. Plasmid. Remember that the plasmid had some genes that were helpful, but not essential. So if you were a microbe and you're going to use catalase every day, all day, that's essential. That's essential for you. Or your enzyme that makes folic acid, that's essential. But having an antibiotic breaking protein, that's kind of a specialized thing. So that's an example of a gene that could be carried on the plasmid. You have to have the DNA instructions to make the protein if you even get a chance to have the protein. So any enzyme that the cell has, they have to have the DNA or the gene to encode it. Hang on just a second here. Do you have a class?
At 1:30, yes.
At 1:30, okay. Someone lingering around out here suspiciously, but they don't have a class until 1:30. So that's okay. Okay. So enzymes, then, if we go back to this picture, there are enzymes hidden in these arrows. So you could write that in. There are enzymes with a S here. There are enzymes here. There are enzymes here. Multiple ones that are doing these processes for the cell. Enzymes that tend to do breakdown processes. Whoops. Pen. Enzymes that are doing breakdown processes tend to require water. So I'm drawing this in here to indicate that water is required for these processes. And energy is produced. Just draw arrows down to there. So when we break down food, this is true for people as well. Big food molecule, breaking it down to get energy out and get our building blocks, water is required. Energy is released. For anabolic processes, it's the opposite. You have to use energy. You have to have input of energy to build bigger things. And when you build those bigger chains of molecules, that tends to release water. I now have bugs flying around me out here. So when you build up, that requires energy and it tends to release water. When you break down substances, that tends to require water and release energy. All of these things that are happening between the molecules that the enzymes are catalyzing, what's really going on is that there are chemical bonds breaking or there are chemical bonds being formed. So if bonds are broken during the process, that's generally catabolic. And if bonds are forming, that process is generally going to be anabolic. Whether bonds are breaking or whether bonds are forming, that requires electrons to be moving around. So I'm going to go back to my camera now. Switch it up. Okay. What I want to do here is to kind of talk about what I need you to know out of this lecture and the next lecture. So you might want to write this down. I'm not going to try to write all of this on my tacky screenwriting right now. But what I want you to know out of this lecture is the information about enzymes. And this type of stuff out of this lecture about enzymes, that's going to be multiple choice, I'm pretty sure, on the next exam. So I might ask you about which one of these is a cofactor, or what else could I ask? Like, what's the definition of an apo enzyme versus holo enzyme? That kind of information is going to be multiple choice. What's coming next is I need you to know some major details about the different styles of metabolism, which are aerobic respiration, anaerobic respiration, and fermentation. So I'm going to say those again so you can write them down. We have three big styles of microbial metabolism that they use to break down food and make energy. Aerobic respiration, anaerobic respiration, and fermentation. And what I tend to do on that is want you to do a compare and contrast. That's the kind of information that's going to be some in the videos that you will watch for Friday. And then Friday's lecture, I really get into the details of those. So that's what's going to be kind of coming next. And the comparison of those three styles and knowing the differences between them and what's similar between them, that's one of the really big things on exam 2. So I wanted to kind of let you know that before we get into this next section. So go back here to the pictures. So this process, all those three processes of aerobic respiration, anaerobic respiration, and fermentation, they're going to start with a sugar. In this class, I teach all three of those processes as if they start with a sugar. And the example sugar is always glucose in these pictures. So this picture is showing the first steps of aerobic respiration. So glucose can be broken down through glycolysis, and then it can go through Krebs cycle, and then it would also go through electron transport chain usually, which is not shown on here. But any of these processes that break down glucose, we can call that oxidation. Oxidation. And depending on how your high school chemistry was, you may or may not have talked about oxidation and reduction. My high school chemistry, I swear, did not ever use those terms because I really loved chemistry in high school, and I knew it really well. But when I got to college and people were talking about oxidation and reduction, I didn't know what they meant. So for this class, I want you to know that oxidation of a food is breaking down of the food. And the name of this term implies that oxygen is needed. Sometimes it is, sometimes it's not. But as glucose is getting broken down, there are chemical bonds in this molecule that are getting broken, and the energy that's coming out of those bonds is captured. Trying to write captured. The energy out of those bonds is captured in little packets. And the energy capturers, you've heard of these before, but where this energy is captured is in ATP, NADH, and another one is FADH2. You can raise your hand in your room if these names of molecules make you feel bad. Like, oh no, I don't want to talk about these. I think that I do a really good job of explaining what the point of these is in those videos that I want you to watch. And I'll talk about them some on Friday as well. But if you've always felt like, oh gosh, I don't even know what NADH does, after this class you will. Because I think when I learned about this, I didn't get it and I didn't like it. So when I go to teach it, I want to do a good job. I always try to teach to rectify what went wrong with me when I was learning. Okay, so kind of to reiterate here, making new bonds or breaking bonds requires electrons to move. So if I have, I'm going to draw a different example here. If I have a molecule that has within it a carbon-carbon single bond, so this would be single covalent. I bet you remember that. Single covalent bond. And it's holding these two carbons together really strongly. And this bond is between electrons. These two carbons are sharing electrons. When that bond breaks, it's going to release energy. So anytime a bond breaks, energy can be released from that. What the cell needs to do is store some of that released energy in a temporary fashion. So we break the bond. This is a catabolic process. We want to get energy out of that bond breaking and store it temporarily in those three molecules that we wrote down a minute ago. So one of those is ATP. You probably memorized that ATP is the energy supply. Money. I tend to think of it like money. Here's some money. $. You can spend ATP to do work. You can spend ATP to do work. Or you can make ATP by capturing energy. So you're going to capture some of that bond energy during catabolism. Like, you take glucose, you break down the bonds, and you capture some of that energy in the form of ATP. There we go. So you can make money, you can spend money. Make money, spend money. Make money, spend money. ATP is the energy currency of the cell. In this class, I don't need you to know how to draw ATP. I don't need you to know how to draw ATP. But the triphosphate part is kind of important. So this over here is adenosine. It's pretty much an A base from DNA, almost. And then it's got on it 3 phosphate groups. 1, 2, 3. 3 phosphate groups. So we call this adenosine triphosphate. There is also a version of this, like I want to hide it with my hand, but that's not showing up to you. So pretend I'm hiding this with my hand. There's a version of this similar molecule called ADP with 2 phosphates. And there's a version with just 1 phosphate called AMP. This would be diphosphate. This would be monophosphate. What I want you to know is ATP is the most energized form. This has the most energy out of these 3. And you probably already know that. The other thing I want you to know is that ATP and ADP cycle back and forth between these 2 versions. So ATP can get broken down into ADP plus a phosphate, or ADP plus a phosphate can get built back up into ATP. I'm going to go backward because I want to erase this stuff. There. I'm going to draw something different here that's really important. Pen. Okay. And it's the same arrows. Okay. So these are going to flip-flop back and forth. And this little drawing you should memorize if you don't already know this. I have a hard time drawing and talking well at the same time. Okay, so cells can take ADP and some energy and use that energy to add on a phosphate to form ATP. ATP has a lot of energy that way. ATP can get spent to release energy and a phosphate and turn back into ADP. So if you've drawn this stuff in here, this side is the make money. This is to make ATP. This would be like we burn glucose to make ATP, and we're storing energy that way. And this side is the spending side. The cell would spend ATP and get some energy out of it to do work. I want to write also, what do cells spend ATP to do? Think about that for a second. What do cells literally do with ATP? Oh. It's perfect timing. Help someone type in a login here. There you go. What class are you going to?
Science.
Science. Okay. That's appropriate. So maybe you thought of some things during that time. Cells can spend ATP to provide motion. It's supposed to say motion. Motion. So when we move our muscles, we're spending ATP to do that. We spend ATP to build. Pretend that says build. Bigger molecules. Build bigger molecules. Like, I'm a gram-positive bacterium. I need to build more peptidoglycan. That takes ATP. We use ATP to copy our DNA. We use ATP to build proteins. We use ATP to divide cells. Try that up here. Divide cells. So lots of different things. Any major task that a cell needs to do is going to require ATP. It is the energy currency of the cell. I'm going to stop there right now because talking about NADH, I need more than 3 minutes or 1 minute or whatever I've got. We will be using this same slide set for Friday, but remember, you need to watch those three videos instead of reading for Friday. That's the assignment for Friday. There is a quiz, and I hope that we will be in person on Friday. I just haven't gotten the test results back for Moses, so I'll let you know. And I also would love to have any feedback that you all can give me about if you had problems seeing, hearing, so on during this team session. Have a great day, and if you want to stay on and talk, I will be on for a while. Okay. Hey, Alyssa, I saw that you came in part of the way through, so I'm going to update that in the attendance. So I need to stop recording.